Good morning, afternoon, and evening to everyone. I think we have members and participants from all around the world, so we are all in different time zones. So welcome to everybody to today's webinar on a critically important topics in global health. Our topic today is harnessing genomics, the future of vaccine and drug development. And I'm absolutely glad to see so many of you joining us today. Malaria remains one of the most devastating infectious diseases worldwide, affecting millions of people, particularly in sub-Saharan Africa. And despite significant advances in malaria control and prevention, challenges such as drug resistance, vaccine efficacy, and evolving parasite strains continue to undermine the global effort to eradicate the disease. In today's session, we will explore why genomic monitoring has become a powerful tool in the fight against malaria, enhancing on our ability to track parasite evolution, assess vaccine effectiveness, and optimize drug developments and drug deployments in all parts of the world that are affected. Through the analysis of genetic data, researchers and public health professionals are now able to gain deeper insight into the mechanisms of resistance, transmission pattern, and immune responses, ultimately accelerating the development of more targeted and effective interventions. Today, we will be hearing from our panels of experts at the Genomic Surveillance Unit of the Sanger Institute, who will share with you the latest research, challenges, and breakthroughs in the use of genomics tool for malaria control. From sequencing of malaria parasite genomes to monitoring the resistance of markers, this webinar will cover a wide range of topics essential for anyone working on malaria research, vaccine development, or global health policy. And I'm really delighted to be introducing my colleague on this call and sharing with you their insights and their experience in this area. We hope that you will find today's presentation informative and inspiring as we continue to work towards a future free from malaria. Thank you for joining us. And without delays, I will turn over to our speakers in a minute. I just want, from a housekeeping point of view, to say this session will be recorded today, so people will be able to access it, share it with others, reflect on the information in it. We've also enabled the question and answer tab, so feel free as we go through today's presentation to add your questions on that we will be reflecting on, answering and interacting with you at the end of the session. As you could see, so colleague Dr. Vicky Simpson, Christina Ariani will both go through their insight. They are familiar to so many of you and will become familiar during this talk to people who have not met them before. And our head of business development and lead that taking the vision of where we move in this area, Janet Houghton, will bring in the call to a close as this. So without any further delay, I pass you on to our first speaker, Christina. So Christina, let's get started.
Thank you, Sahir. I think I'm first, actually. So just to introduce myself briefly. So my name is Dr. Vicky Simpson, and I am the Head of Partnerships and Community of Practice at the Genomic Surveillance Unit at the Wellcome Sanger Institute. And I've been working in malaria genomics with the Malaria Gen Network for the past 15 years. So my role is principally around global engagement. There's a particular focus on generating, curating, processing and analysing malaria genomic data across human, parasite and vector communities. And a major output of this collective effort is large, curated, harmonised, open data resources that are powerful tools to answer public health questions and for research and development. Before that, my background is actually in pharmaceutical science, chemistry, drug formulation. So my plan is to draw on both of these knowledge bases to provide a global perspective on malaria, disease control and elimination. So while the scale of the issue of malaria is global, if we look at the 2023 WHO malaria report, we can see that the impact is very disproportionate. The majority of the 249 million cases are within the African region, so that's the red circles. And there's considerable diversity of impact in Africa. From the size of the circles, it's really clear that a really small number of countries have the highest burden. And while this graphic gives a sense of the diversity of impact of the disease in terms of number of cases, we also need to consider the variation in disease as well. So again, using data from the WHO malaria report, but bringing in also information from the scientific literature, we can see the global variation in species distribution. So using the same color codes and regions from the previous slide, what we have here is a breakdown of the cases and deaths by region, and also the species distribution, the approximate species distribution in those regions. So the African and Eastern Mediterranean regions, which are in red and blue, have the largest number of cases and deaths, and they're also predominantly Plasmodium falciparum. So this is one of the reasons why often falciparum and Africa in particular are the areas of focus when it comes to malaria control and elimination efforts. However, it is important to note that even if we are just going to focus on falciparum and Africa, we can't ignore the impact of what is happening in other regions on that area, and neither should we ignore those areas themselves. Each of these has its own genomic profile. There are a significant number of cases arising from Plasmodium vivax, malariae, ovale globally. And given that many malaria control interventions target P. falciparum almost exclusively, depending on their success in eliminating this parasite, the landscape will look very, very different in the next few years. Having mentioned malaria control and elimination efforts, it's worth briefly highlighting the three main strategies, which are drugs, vaccines, and insecticides. So the frontline drug treatment for Africa is artemisinin combination therapies, or ACTs. We will be using that abbreviation as we go on. So artemisinin is a fast-acting endoperoxide, and in ACTs, it's combined with a drug of another class. So an example would be partnering with lumefantrine, which is an aryl amino alcohol. But as there is emerging resistance to ACTs with two drugs, triple artemisinin-based combination therapies are now being explored to address resistance, offer a broader spectrum of action, and reduce the risk of treatment failure. As part of its call for action to preserve malaria control gains and ensure the ambitious 2030 elimination targets are met, WHO has approved additional interventions such as RTSS and R21 vaccines and new generation insecticidal nets with dual active ingredients. For both of these interventions, they are being deployed at scale and against a background of emergence and spread of partial resistance to artemisinin resistance, insecticide resistance, and invasive vector species. So I personally find the vector space also incredibly interesting, but that will be a subject of a future webinar. And for now, I will focus on the drugs and vaccines that target the falciparum parasite. So picking up on what was said previously around drug resistance, here we are showing the history of the introduction of principal antimalarials and the first emergence of resistance in the field. So the colours of the bars show the chemical class of drug. So artemisinin, which is an endoperoxide, is a blue bar, and it's shown as a monotherapy, which is a single bar, and as a combination therapy, so as a double bar, as is the case when it's partnered with lumefantrine. So in this case, the second bar is pink, as it's an aryl amino alcohol. And the triangles show the point where resistance first emerges in the field. So we can see that resistance has occurred for many of the principal antimalarials in use today. So opportunities in drug development include tackling parasites that are resistant to artemisinin, reformulating existing drugs for malnourished and low-weight infants, and tackling co-infections, so considering P. falciparum alongside Vivax, malariae, or ovale. And later on, Christina Ariani will address some of these elements in the Harnessing Genomics section of the webinar. If we consider the vaccine space, so as I mentioned previously, so this has recently been approved by WHO for mass rollout. And here we are showing the deployment plan for RTSS, which is one of the approved vaccines for 23-25. And 18 million doses have been allocated across 12 African countries. Now, the framework calls for doses in children from 5 months of age, and each child would receive 4 doses. So while 18 million doses has been allocated, as each child would require 4 doses, it's clear that this is insufficient to meet the needs of over 25 million children born each year in regions with moderate to high malaria transmission. And just kind of looking at both RTSS and R21, which has also been approved, both of the vaccines target a protein found on the surface of the sporozoites, which helps the sporozoites invade the liver cells. So while it's great news that these vaccines have been approved and are being rolled out, the dosing is challenging, each requiring 4 doses. And both of these vaccines have shown that efficacy is improved using a seasonal approach for rollout. So running public health campaigns to deliver vaccines seasonally is much more complex to implement in practice compared to an age-based approach. So therefore, it would be reasonable to expect that the efficacy will be negatively affected if it isn't delivered according to this framework. So I'm just going to try and draw this global perspective to a conclusion before I hand over to Christina. And I'd like to leave with this thought. So there are considerable opportunities for malaria drug and vaccine development. There were 249 million cases in 2022. We are currently awaiting the WHO malaria report. It should be out in the coming days. But even as we strive towards the ambitious elimination targets, the burden we know is still great. There are issues with current interventions. We do not have all of the tools in the toolkit that we need in order to eliminate malaria. Drug resistance is emerging, and there are challenges around vaccine delivery and the impact on its efficacy. Genomics is a really powerful tool that can be integrated into drug and vaccine development. And for that part of our webinar, I will hand over to Dr. Christina Ariani.
Thank you very much, Vicky. Good evening, good morning, good afternoon, everyone. I am Christina Ariani, and I lead the malaria parasite surveillance team at the Wellcome Sanger Institute, the Genomic Surveillance unit. I have been working on malaria for the past 10 years at the Wellcome Sanger Institute, and I've been trained on evolutionary genomics of mosquitoes and malaria over the past years with my PhD at the University of Cambridge. But today, what I'm going to explore with you is the relevance of genomics on the development of malaria vaccines and drugs. Yeah, so what we're going to be exploring today is genomic technologies and its application in malaria. So the rise of genomic technologies has prompted incredible advances in our understanding of the biology and evolution of both malaria parasites and vectors. The research, as a consequence, has considerably helped our understanding of the mechanisms of resistance, genetic diversity, and adaptations that drive malaria persistence. As Vicky said, you have seen the map of the malaria persistence in the world, and is still very much of a problem. But a considerable portion of the current genomic knowledge of malaria parasites and vectors has been generated by our teams at the Wellcome Sanger Institute. And just a sec. Sorry, yeah. And we have decades of experience in malaria parasite genomics, and we continue to innovate for scientific and public health impact. So when we combine our senior members of staff, there is more than 100 years worth of experience in this field. And with all that experience, we've built the largest, most geographically diverse, and most accessible malaria genomic data resource. This resource contains more than 38,000 whole genome sequences of malaria parasites from 38 countries. And as you will hear at a later point, that a very similar resource is also provided for vectors. The data is freely accessible and open source, so that means that anybody with an internet connection can access that very rich data set. But we're not focused only on those resources. In addition to that, we also innovate in lab techniques and bioinformatics analysis. And because of that expertise, what I'm going to be addressing today is what is the problem that we are facing now with malaria and how genomics can help. As Vicky mentioned in her presentation, there are records of artemisinin therapy failures. Here's a cartoon of the current state of play of resistance to artemisinin. It's worth mentioning before I go ahead that what we mean by resistance here. So when we have an artemisinin resistant sample, that means that parasite has a delayed clearance, just to clarify. And the artemisinin resistance has first emerged in Western Cambodia early this century and have since reached high levels across Southeast Asia. As you can see on the map there, there's currently a high prevalence in Southeast Asia. There have been sporadic reports elsewhere around the world, and while there is little evidence of resistance in West Africa at present, the situation in East Africa is particularly worrying. So if we look at where artemisinin resistance is occurring, perhaps there is no surprise that this is areas of greatest artemisinin combination therapy use. As artemisinin combination therapy, as Vicky mentioned, we're going to call it ACTs for ease, are used more across the continent. The emergence and spread of resistance is likely to accelerate. In the map here, we show in red Uganda, which is a country with high records of artemisinin resistance, and in orange, the other countries which have recorded medium rates of artemisinin resistance. It's worth noting that WHO has validated 13 molecular markers, and there are 9 candidate markers associated with resistance to artemisinin at the moment. But what's really interesting is that the 5 most frequent markers in Africa are rare elsewhere in the world, suggesting the emergence of artemisinin resistance in Africa was independent from previous emergence in Southeast Asia. Right, so the real problem here is that ACT, the artemisinin combination therapies, they fail when the partner drug fails, not when artemisinin fails. For example, ACTs remained effective in Southeast Asia long after artemisinin resistance emerged. And it was only when the treatment, when the drug, the partner drug, piperoquine in that case, failed, that the whole ACT started to failing. The situation that we are now is that the most commonly used ACT in Africa, which is artemether lumefantrine, is becoming, there are records of it becoming resistant and becoming less effective. Therefore, reliable genetic markers would allow better monitoring of the rise of resistance and timely action to be taken to change frontline drug policy. However, one problem that we currently have is that such genetic markers don't exist, and we really need to find the genetic markers that determine lumefantrine resistance in Africa. So, what do we predict is going to happen? To answer that, we gathered data from 112,000 published genomes to look at the spread of artemisinin resistance globally. From the data, what we could say was that at the time of discovery of resistance in Asia in 2008, resistance markers were already at high frequency in several countries, as you can see there on the plot. They then rapidly increased in frequency across Southeast Asia, reaching near fixation in around 10 to 15 years. And on the plot, all the blue lines are the Southeast Asian samples, so you can see that. But what's important is that what we saw in Southeast Asia in 2008 onwards is now occurring in Africa, but delayed by around 10 to 15 years. And on the plot, you will see the yellow brownish lines represent African countries. And you can see the slope of the lines are relatively similar between the African countries and the Southeast Asian ones. So in our review, we discussed how dangerous this situation is because we are likely observing the start of a major spread of resistance there, and there are limited options should treatment start to fail. If you want to learn more about this paper, you can get the QR code there, or you can find it on bioRxiv at the moment. So as a result of the urgency of the emergence of artemisinin combination therapy resistance in Africa, several major players in the field launched a call for action. And this is that document here that you can see on the screen. So in this document, they're calling in multiple donors, including philanthropic foundations, private sector, and others to join in the call to make alternative combination drugs available and more affordable. So now I'm going to change gears slightly, and I'm going to just talk to you about vaccines, because drug resistance is not our only interest. We are interested in the vaccine rollout as well. And then here I'm showing the countries which already have plans for RTSS vaccine rollout. As Vicky mentioned in the beginning, we have two major vaccines that have been we have two major vaccines that have been deployed that have been approved by WHO, and here I'm showing RTSS. So we want to know with genomics what effect the rollout of vaccines will have on the epidemiology of malaria, for example. And as another example, will we start seeing vaccine escape mutations? Will we start seeing an increase in other malaria parasite species in Africa? So, and the reason for it is as the vaccine target Plasmodium falciparum, will other Plasmodium species fill the gap and become more prevalent? So these are just some examples of questions that genomics can help address. And to ensure the ambitious targets set by WHO by 2030 are met, we should leverage each other's expertise. From the public sector, private sector, academia, and us, and maximize gains in malaria control. So genomics can help with tools that inform strategic decisions by key stakeholders to inform development of new methods of malaria control. And here we'll address some of it. One example that I'd like to bring to your attention today is how genomic data can help on the development of new products for malaria control. Here I show a simplified and generic pipeline that can be applied to drugs and vaccines as examples. In the next few slides, I'll focus mostly on these three areas of the development pipeline: the research and development, clinical trials, and post-market surveillance. So focusing on the R&D bit first, the reverse genomics. So reverse genomics has significantly advanced the field of vaccinology, so reverse genomics is a strategy used to identify potential vaccine candidates by analyzing the genome of a pathogen rather than through traditional methods like isolating and culturing the pathogen itself. With that reverse genomics, it's possible to predict antigenic targets and epitopes, for example, and bioinformatics tools can be used to predict which proteins are most likely to be recognized by the immune system. The malaria parasite Plasmodium falciparum is a highly complex organism with a very large genome that is highly AT-rich. The parasite evolved incredibly well to successfully fight the immune system, and because of that, it's very difficult to develop a new malaria vaccine. But genomics of both the parasite and the human immune system may help a lot in developing a new effective vaccine. And due to our vast experience in analyzing tens of thousands of parasites' genomes and to our deep understanding of the natural variation of parasites in the field, we are really well placed, actually, to collaborate with vaccine development teams to figure out how we can leverage from the very large data resource that we have to develop new targets for vaccines. The next part that I want to bring to your attention to is on clinical trials. Clinical trials for malaria cover a broad spectrum of research with the ultimate goal of eliminating malaria. These trials often take place in malaria-endemic countries, and the results can have a significant impact on the development of new drugs, vaccines, and strategies to reduce malaria transmission. With the threat of drug resistance and evolving mosquito populations, ongoing clinical trials are essential for staying ahead of the disease, as you can imagine. And one type of clinical trial that I'm particularly interested in, and I'm going to be focusing my attention right now, is the treatment efficacy studies. These are studies to test whether a drug remains effective or not. And combining whole genome sequencing and a TES, we call it the treatment efficacy study, we call it TES. So combining whole genome sequencing and TES are of major importance right now. That's because we need to find a molecular marker that confers resistance to lumefantrine, as I mentioned right at the beginning. And the most effective way of achieving that is sequencing samples with a resistant phenotype and compare to samples with a susceptible phenotype. And then bioinformatically, we can take those cohorts and identify the marker of resistance. This is very important right now because then we can track resistance strains and public health authorities can make informed decisions and manage malaria cases locally. So this is an area that I think the malaria community has their eyes on, which is to find the molecular marker for lumefantrine. Next slide, please. The last part that I wanted to talk to you about is the post-market surveillance and how genomics can help on that area. So genomics can play a significant role in the post-market surveillance of malaria drugs by providing insights into the genetic factors associated with drug efficacy, resistance, and patient response. We can, through genomics, monitor drug resistance, any evolutionary changes or selection in the parasite genome as a result of the implementation of new drugs or vaccines. So, for example, there is a new drug that just came out of the market and is deployed somewhere. We can monitor together those samples to understand how they are impacting the parasite population in the field. And of course, any post-market surveillance, we can monitor any treatment failures as a result of the deployment of those drugs, including the identification of new molecular markers that could emerge as a result of implementation of a drug or a new vaccine. So to finish and conclude my section, the result, yes, thank you. The result of collaborations between the malaria genome scientists, the public and private sectors can have significant impact on WHO global policy practice guidelines. And the data generated could impact on national and global procurement and rollout strategies, because if a drug is no longer effective, it's going to impact the procurement. Understanding how long frontline drugs and vaccines are likely to be effective will have significant financial implications on global research funders and the private sector with respect to portfolio and pipeline investment. So I just wanted to leave you with that thought that understanding what is happening in the field has several implications from procurement to policy and to actually containing the disease and saving lives. So with that, I'll hand over to my colleagues. Thank you very much.
Thank you, Christina. A tour de force as always. So we have a few questions in the Q&A. Please feel free to add more. But so I'm going to try and moderate this and also contribute to the answers. But so the first question we have is, what can be done to minimize the skills and capacity gap as far as genomic surveillance in Global South is concerned? So I obviously have some thoughts on that, but Christina, would you like to take the first swing?
Yeah, sorry. Can you repeat the question, please?
So, what can be done to minimize the skills and capacity gap as far as genomic surveillance in the Global South is concerned?
Well, that is an excellent question, isn't it? So I think training is the decentralization of genomics has to be taking place as a matter of urgency, and this is happening right now. And then the training that comes along with it is also very important. And it's the training not just on the laboratory skills, but also on the analysis skills and the interpretation. So I think the Global North has the obligation to work towards making sure that, you know, the malaria endemic countries are well equipped, partners are well equipped, and we can support how that can be done in country the best possible way. Vicky, I don't know if you have anything to complement.
Yeah, so I think that we have been committed to training and capacity strengthening for since as long as I've been a part of the organization. But I do think that what's really wonderful now is there are so many more African-led initiatives than there were 15 years ago. Thank God, you know, funders have transitioned to the, you know, a new model where they're actually, you know, they're empowering the leadership from malaria endemic countries, which is how it always should have been. So there's the African Bioinformatics Institute that's been announced this year and is going to go from strength to strength. So we're expecting to see wonderful things from them in 2025. Africa CDC PGI also has a mandate around genomic surveillance for many diseases, but also for malaria. We were actually involved in kind of contributing to their roadmap, which should be launched very soon. And I think both of these major initiatives are interested in what can be done to kind of close the gap. Importantly, from my own personal perspective, there are many different parts to this. You know, genomic surveillance is a massive area. And I think when it's not possible to address all of those things all at the same time and at scale. So I think the key thing for me is to really identify where the most impact can be had with the resources that are available. And so from where I see it, where I think what we should be doing is working with players like the African Bioinformatics Institute, like Africa CDC, PGI, and connecting in to what they are trying to achieve in a meaningful way. So I'll move on to another question. So we also have a question around things with the most potential. So Christina, of the things that we've discussed, which is the area with the most potential in your opinion?
Yeah, thanks, Vicky. Thanks, Rachel. That's a really, really good question. I think the vaccine is a very interesting and exciting moment because it's something that we've never seen being deployed before. So it's something new, and it's something that we should be monitoring very closely to ensure that the effectiveness remains. But I'm also extremely interested in looking at treatment efficacy samples to be able to find a marker for lumefantrine resistance. I think the whole malaria community is very interested on that field, because unlocking that molecular marker for lumefantrine resistance will mean that we can then monitor it. At the moment, we can't monitor it because we have no genetic marker for it. So I think there are two sides here. I think one is monitoring what is happening, but the other one is a little bit more urgent to find the marker.
Thank you, Christina. So in the interest of time, I will hand over to Janet to comment, to make any final closing comments.
Yes, sure. Thank you. And thank you, Vicky, and thank you, Christina, and thank you, Sahir, as well, for the introduction. I think just to wrap in the final question about when is the vector webinar, that will be in the new year. So we will advertise it widely, and please do let colleagues know who might be interested in that. So I think, as you have heard today, we've covered a wide range of topics that are absolutely critical, from the role of genetic monitoring in tracking the malaria parasite evolution, to how the really important data insights can shape the development of more effective vaccines and drugs. And it's clear that while significant progress is being made, and I think we should keep that point right in front of us, there's still very much left to do in the fight against malaria. And so as we bring this short session to a close today, I want to repeat our strong commitment to making sure we play our part in the global effort to control and eventually, hopefully, eradicate malaria. And we intend to do that by deploying this considerable expertise, those years and years of expertise, the data insights that we're able to generate, and the active engagement, so that we can, with you, create really strong, effective countermeasures to advance our understanding, advance our research, and improve the tools that are going to make a difference. And that's why we're all focused on this topic. So we continue to focus on genomic monitoring, and obviously that's intended to make sure that we all stay ahead of the emerging challenges, you know, the drug resistance and evolving parasite strains that you've heard discussed this afternoon, and to make sure that we can have more targeted and adaptive interventions. And so the key to this, though, is linking together, joining together, bringing together those islands of expertise, working across sectors, across borders, across disciplines, to make sure that that fight against malaria is a priority, and it's one that is informed by the latest science and data. So we look forward to continuing this journey with you. We'd like to invite you, please, to stay engaged with us. We'd like you to add your perspectives and insights. Please do feel free to put in follow-up questions, to contact us if you would like to discuss anything one-to-one. The aim of this, as I say, is to, with us all working together, ultimately eradicate malaria globally eventually, and not too soon in the future, hopefully. Thank you for your time and your engagement today. The recording will be shared, as will the questions and answers, and so please stay safe, stay engaged, and let's keep pushing forward in this absolutely essential work. So thank you to speakers, thank you for joining us. Goodbye.